Welcome back, everyone. For this week, we'll be performing the polymerase chain reaction. So this week's lab will actually be the very first lab for our semester-long project. So what we want to do this semester is try to purify the protein beta-galactosidase so that we can analyze its function and see how it works. So here I just have some key information that'll be useful for this project going forward. So here you can see in this image that beta-gal is actually a tetramer, meaning it is a complex made of 4 small monomers. So one monomer, or one DNA strand of beta-gal, is about 3,075 base pairs long. When we look at the protein and its molecular weight, we get about 456 kilodaltons for the entire complex, or 114 kilodaltons for a single monomer. And so the function of beta-gal is to hydrolyze the cleavage of beta-galactosides into monosaccharides. So these are sugars like lactose that can be broken down into their monosaccharides, glucose and galactose. So for us to study this protein of interest, we have to kind of start off at the very beginning, which is looking at its DNA. And the best way to do this is to make tons and tons of copies of this DNA so that we can progress further with the experiment. So the best way for us to do this is by using a technique known as the polymerase chain reaction, or PCR. And what it does is it copies a specific region of DNA and amplifies it in vitro. So this makes it very simple to use in a lab setting. It relies on a very thermostable DNA polymerase, or for this case, a Taq polymerase. And for it to work, it also requires DNA primers that are designed very specifically for our DNA region of interest. So the way that it kind of works is the reaction is repeatedly cycled through different temperatures. And as these temperatures change and as these cycles continue, we produce many copies of our DNA target. So PCR has been used in many practical applications, from in the lab as DNA cloning to medical diagnostics as well as forensic analysis. So like I said, PCR is used to make millions or even billions of copies of target DNA that we can use further along into this semester project. Many copies of the DNA is necessary for further experimentation, such as using it for sequencing, or DNA gel electrophoresis, or cloning it into a plasmid. So when we set up a PCR reaction, there are only really 5 components that we need. The first one is obviously our template that contains our gene of interest. And this could be just our gene of interest. It could be a plasmid containing our gene of interest, or even a longer strand of DNA that has our DNA, or has our gene of interest somewhere inside of it. Ideally, though, we would prefer just the gene, as it'll make the chance of contamination less likely. In order to actually specify our gene of interest, we'll need to include primers. So primers are short strands of DNA, usually 15 to 25 base pairs long, that attach to the 5' and 3' ends of our DNA strand. Once these primers bind, the DNA polymerase is able to locate these primers and are able to replicate the whole gene. In order for this replication to occur, the DNA polymerase needs free nucleotides so that it can actually build the entire DNA strand. And for it to function properly, it also needs to be in a specific kind of buffer, as well as have other cofactors. So here I just kind of want to give a little example of what I mean by the primers. So up here we have our template DNA. When we break it apart, you can see that we have 2 little small red strands of DNA. These are our primers. Primer 1 is our forward primer, and primer 2 is our reverse primer. So for today's lab, or for this week's lab, you're going to have to write a lab report. And in this lab report, I want you guys to talk about the primers and even design the primers for beta-gal. So to kind of give you a little nudge in the right direction, what I want you guys to know is I'll be giving you I guess the entire map or the entire DNA strand of beta-gal, and it's going to be written in the 5 to 3' direction. So what this means is that when you design your primers, the forward primer will always be the easiest because it's literally the copy pasta of whatever is on the 5' to 3'. So you can see that it's exactly the same. For the reverse, it is going to be the reverse strand. So you're going to have to actually find the other base pair that connects to whatever is written there. And then when you write it down in your lab report, you're going to need to flip it. So this means that when I see it on your lab report, it should be the reverse complement. So here you can see that the primer is going in the opposite direction. So when you write it down, I want to see it as, I guess, like T, C, A, T, A, G in that order. So that is how you design your forward and your reverse primer. If you have any questions on how these primers should be designed, please email me as soon as possible, and we can talk about this. So just going a little bit more into the PCR. So here we have our template DNA. The template DNA is broken so that the primers can bind. And once the primer binds, the Taq polymerase is able to bind to the primers and extend the DNA. So we can do this over many, many cycles. So you can see after 1, we have 2. After 2, we have 4. And after 3, we have 8. So we are able to grow the amount of DNA exponentially. So you can see from, I guess, that little basic summary that there's 3 key steps of the PCR reaction, or the PCR. So in the very first step, we have a denaturation at 96 degrees Celsius. So at this heat, the double-stranded DNA, or the hydrogen bonds of the double-stranded DNA, are able to break, and this gives us our single-stranded template. We then lower the temperature to somewhere between 55 to 65, depending on the length of our primers. And at this cooler temperature, the primers are able to bind to these regions of the single-stranded DNA. Once the primers bind, we raise the temperature back to 72, and then our Taq polymerase is able to function and reproduce the entire region of interest. So just kind of showing it again, we have our template strand, we denature it at 96 to make 2 single strands. We then lower it down to somewhere between 55 to add our primers. Set the temperature to 72, and then we have the strand, the DNA being extended. So once it's fully extended, we'll have 2 complete sets of DNA, and that will be 1 cycle. We then start it all over again, and you can see how this is able to make an exponential growth of DNA. So you can see this in your lab manual, but I just kind of want to put it here for you guys to kind of look at. So these will be the conditions that we use for the PCR reaction. So again, we'll actually start off with an initial denaturation at 98 degrees Celsius, just to make sure that the gene template we put in there is completely single-stranded. And then after we have that initial denaturation, we'll have that 25 to 35 cycles. So here's our initial denaturation, the annealing temperature, and then the extension. Once this happens about 25 to 35 times, we'll have a final extension at 72 degrees for 2 minutes. And then at the end, we write this hold at 4 degrees Celsius. And the only purpose of this hold is for if you happen to go out to lunch and you're just waiting for the thermocycler to kind of run, You just want to make sure that the DNA isn't somewhere cold when you come back. So once we're done with the PCR, we need to kind of make sure that we actually got our gene of interest, because technically we won't really know. And the best way for us to do that is by using gel electrophoresis. So the DNA can be run through a gel matrix. For this, it will be made out of agarose. And depending on the concentration of agarose, we can actually control how far the DNA goes through the gel matrix. So think of it like this. If the agarose is more concentrated, the matrix becomes a lot less permeable, so the DNA will move slower down the gel. If the agarose is less concentrated, that means our gel will be more diluted. So this will make it easier for the DNA to go through. So we also run a DNA ladder alongside our PCR product to kind of see the amount of base pairs that our DNA strand is. So the way that this gel electrophoresis works is by utilizing the negative charge on the DNA. So remember that DNA's phosphate backbone has a negative charge to it. So when we put this in an electric field, the negatively charged DNA is able to move towards the positively charged poles of the electric field. So this is how we're going to get our DNA in both the ladder and our PCR product to move down the gel. So here's kind of a little example of how this would work. In one lane, we have our DNA ladder that we know the sizes to. And in the second lane, we have our PCR reaction. So when we run these two side by side, we're able to see that here our PCR, after this PCR reaction, we got about a size of 400 base pairs for our DNA. If that's what we're looking for, then that's great. But obviously for us, beta-gal is much bigger than that, so this is not what we want to see. So I hope that helps you guys understand a little bit about the polymerase chain reaction and how DNA gel electrophoresis works. If you have any questions about either of these subjects, please feel free to email me at any time.